So, without any further delay, it's my pleasure to introduce Dr. Maria Mota um, from Portugal. Dr. Mota is a distinguished biologist and certainly a very influential leader in the scientific community. She graduated in biology and obtained her master's degree in immunology at the University of Porto and her PhD in parasitology from the University College London in the UK. Um, she became a leader of the malaria unit at the Institute of Molecular Medicine, IMM, and a professor at the medical school at the University of Lisbon, and became the executive director of IMM. She's also a visiting professor at the Harvard School of Public Health. Recently, she's taken on the role of CEO and I apologize if I do not pronounce it correctly, Dr. Mota, at the Gulbenkian Institute for Molecular Medicine, GIM, a new institution that results from the visionary merger of IMM and the Instituto Gulbenkian de Ciencia, IGC. Dr. Mota has made significant contributions to the understanding of Plasmodium parasites and their interactions with hosts. Her pioneering work includes identifying host molecules that determine resistance or susceptibility to severe malaria and those affecting the liver stage of Plasmodium or interactions between blood stage and liver stage. She's had many accolades over the years. And I'm not going to go into them all. She's a Howard Hughes Medical Institute International Investigator. She's been honored at the national level with Commander Crosses, Gra Cruz de Ordem do Infante de Henrique by the Portuguese presidency. And to date, she's published over 140 articles in significant international scientific journals and holds 5 international patents. As an aside, Dr. Mota is a passionate advocate for women's rights and fair education opportunities, so important to all of us at BSI. She contributes a monthly opinion column to the Portuguese newspaper Expresso, furthering her vision of a knowledge-based society. So without any further delay, I'd like to invite Dr. Mota and thank you again for being our open keynote, and over to you.
So thank you so much, and thank you to everyone that is listening to us. Thank you so much, Dr. Fish, to introduce me in a so kind way. So I'm going to talk about malaria, but I'm going to talk about malaria in a stage that in fact is still not malaria, and I will explain a little bit. So let me just share my, I think it's this one, share. Can everyone see my presentation?
Yes,
Just
we can.
one slide, or you see 2 slides? Just one slide?
We see both slides.
Okay, like this?
Perfect.
One.
Single slide. Yep.
Okay, perfect. So basically, thank you so much again. As I said, I'm going to talk about malaria, but in fact, it's something that happens a little bit before. And I'll come up again to this beautiful image. But malaria itself is a disease that is caused by Plasmodium, eukaryotic parasite, a protozoan, and that this parasite, when it infects red blood cells, every 48 hours, in the case of Plasmodium falciparum, that is the most dangerous form of malaria for humans, basically each parasite replicates in 20-30 parasites. The majority of humans survive with this and they are able somehow to clear the majority of the parasites, but there is a proportion of human beings and children that lives in endemic areas are the ones that suffer the most, that are unable to control the inflammatory response, and they die with this. And our lab and many others around the world have studied obviously this stage that is called the blood stage of malaria parasite, and is the most important as perceived as that because it's really the one that causes disease itself. But prior to this, the parasite needs to be transmitted by a mosquito. And the parasite lives in the salivary glands of the mosquito and is transmitted into a stage that cannot infect directly red blood cells. So in mammalians, in humans and other mammalians, basically the mosquito deposits parasites in the skin, and the parasites must find ways of reaching the liver. This is an obligatory step. And in this liver stage, the parasite really goes from one parasite into thousands of new parasites, and now these new parasites are at a stage that now infect red blood cells. So, what I'm going to talk about is really this liver stage of infection. And we really think is important. And I'm going just to highlight a story that, in fact, started more than 25 years ago, more than 35 years ago, if we start really to look exactly at the dates, and is having now a real impact. And I think this is also good for students, postdocs around the world to see that sometimes things take time, and we just need, not to lose sight of it. Okay, so how I got interested. I did my PhD in blood stages, so I did my PhD working on this stage of the malaria, but then I moved to New York to work with Victor Nussensweig lab. But after 2 months of being there in the Department of Parasitology, I met Professor Vanderberg, and he was already, older senior professor responsible for the parasitology course for the medical students, and he was asking me and inviting me to give some classes. And the first time that I spoke to him, he showed me several different aspects that he was studying. And Professor Vanderberg was really well known for having discovered the ability that parasite has to move in substrate. So the parasite, when is the stage that really is deposited in the skin from the salivary glands of the mosquito, really has this capacity of what we call glide motility. So of gliding in substrates and leave behind a kind of a coat, and is what we can stain and see these beautiful trails. So when I was talking to him, he was showing me several movies that allow him to show how this parasite move, etc. And he showed me this. We were on April 99 when we had this conversation, and he showed me a movie that he has done 10 years before, as you can see here by the date, in 89. And basically this shows the parasite moving, approaching a liver cell. This was in vitro, so this was a hepatoma cell, and it was approaching this cell. And we were just talking, and suddenly the parasite came out of this cell. And I was surprised. I was not surprised by the parasite getting in. That is what we have. We knew from the textbooks, but then we thought, okay, how the parasite really comes out? The parasite has encountered a cell that really is the cell that needs to invade, should stay inside. I asked him, is this really some sporadic? I said, no, I have seen it all the time, because he was doing all the time these movies for showing the parasite how it moved, and really saying we were seeing all this all the time. And I was very surprised about that. But truth is that I didn't know exactly what to think about this. But next day, I met another person that I never met before. Okay, and now I'm having problems too. Okay, sorry. I met another person, Ana Rodriguez, and Ana Rodriguez was a very young group leader that have just joined the department 1 month before. And so we met by a coincidence, and we started, not a coincidence, we were working on the same department, but we met, and then we started talking about it. And I asked her what she was doing, and she said, oh, I work with trypanosoma because trypanosoma, and I'm interested on cell wounding. And cell wounding is this capacity that cells have of sometimes suffering some kind of injury, make holes, and then be able to reseal it. And she said, I work on trypanosoma cruzi, that is a parasite that causes Chagas disease in South America. And I work on this because the parasite really likes to cause these wounds. And I'm really interested to understand the importance of these wounds for parasite life cycle. What are the consequences of that and everything? And I said to her, oh, I think the malaria parasite also makes some wounds. And she said, oh, no, it's not, I'm sure if indeed people would have said before. I said, yeah, but I saw this movie before yesterday, and basically I could see the parasite going in, but also going out. And in the process of going out, special on these aspects here, you really could see some kind of cytoplasm, some kind of material coming out. And so she got curious about it and decided that, we could go two together to talk to Jerry Vandenberg and ask him really if he could show us the movies again. And it was obvious. We saw this movie, we saw many more, and it was seems obvious that parasite, especially when it was coming out, we could see kind of material from cytoplasm coming out. So there was a kind of a wound. So Anna and I and myself, we became interested, and we didn't even say anything to anyone in the department. We just start to work together, and trying to see does really this parasite make some holes on cell or not. And basically, taking a very long story a long time ago, in just one slide, basically what we show is that indeed the parasite, prior to fixing one final hepatocyte, it really traverses several host cells and traverse several host cells by really, making a hole, being inside of cytoplasm. And this cell here, the parasite is inside of cytoplasm and doesn't have a parasitophorous vacuole membrane. And in the final one, the parasite really causes an invagination of the plasma membrane and resides into a vacuole. So this was the way that we had to distinguish. These ones really, we could see PVM, the other ones we could not see a PVM. And so we really were thinking, okay, so this is something very interesting. Of course, at that time, and after 2 years of many experiments really proving that this was true, we published this paper in 2001, and we gave several hypotheses why this was important. The main one is that probably they need this ability to migrate through several barriers because, as I mentioned, the parasite really is deposited in the skin and then basically needs to reach the liver. So probably needs to have this capacity to reach in the skin blood vessel, in the blood vessel to stop in the sinusoids and then be able to pass to the other side to the parenchyma cells. So we really thought, okay, so this seems to be really interesting and cool. And we start to work on this. What could be also, does the parasite also look to specific hepatocytes? Does this activate somehow the parasite for the parasite to sense the environment where it is? All these aspects were being studied. And in fact, it was not just us. We really opened a new field of study, and there is hundreds and hundreds of papers now where people show different aspects, and sometimes new things arise and we don't even notice. After all these years, 2 weeks ago I was interviewing someone to come to work to our institute, and basically this guy was describing this paper here, and basically this paper just shows that while parasite traverses, this causes a huge actin cytoskeleton rearrangement within the traverse cells, and they think this is really preparing the cell to really receive the parasite later on. So it's really something very interesting and very recent. To be so really these came and start to be part of the life cycle of the parasite. So the plasmodium has this capacity of cell traversal, and basically it's a journey across biological barriers. The parasite is deposited in the skin. It's to cross several barriers in skin, reach a blood vessel, then travel to the liver. In the liver sinusoids is stopped, and then it's to cross several to the other side, parenchyma, and there we also know that traverses several hepatocytes. I should say that although we discovered this, 25 years ago, I stopped to work on it. And the last one that we work is that we show that seems that there was some level of activation of the parasite while migrating through cells. But many others start to work on this, and I start to work on a different aspect. So when I started my lab, I decided to focus on this really special aspect that is why hepatocytes, okay? And what are the hepatocytes and liver have so special? What I mean with that? So the parasite reach then a final hepatocyte. It really invades with invagination of the plasma membrane, forming this parasitophorous vacuole membrane and resides within this hepatocyte. And within this hepatocyte, each parasite gives rise to 30,000, 50,000 new parasites, and in just a few hours. So really, this is something very special. We know now from work done by Fotini Sinis and Robert Menard that some parasites get stuck in the skin. They can even invade some skin cells, but the parasite replicates probably in 20, 50 different parasites. So there is something special about these hepatocytes. And it's not only hepatocytes, it's the hepatocytes in certain environment in the liver. Many years ago, in 2010, I really moved to MIT for a few months to do a sabbatical in a collaboration with a very interesting bioengineer, Sangeeta Bhatia. And during that time, we really tried everything. Our lab was very much interested on developing ways of growing a human liver outside of humans. And so we were using all these tools that they have, 2Ds, 3Ds, organoids, this and that, for parasite. And yes, the systems are very interesting. We are now working with them, special because we can work with Plasmodium falciparum, that is the parasite that affects humans, instead of using rodent models. But again, they never replicates in this huge amount of new parasites. So there is something special about the liver. Within this big question, and we have been working many different aspects and discovering many different aspects, but there was something that also was very puzzling. So this liver stage is called frequently the silent stage of infection. And the reason why is that is because, as I mentioned before, this is a stage that there is no symptoms around it, so is not associated with disease. And more than that, there was this concept that when we look to these parasites, and in the liver parenchyma, this is really a parasite that is fully replicating in a liver parenchyma of a mouse, you really see that is untouched. Seems that parasite is never recognized. So people also start to say that it's silent because, basically the immune system would not recognize the parasite when it's, in natural infections. The truth is that we know that this is not always the case. Sometimes, in fact, we can see parasites that do not look so healthy, and they seem to be surrounded by, kind of immune cells infiltrates, macrophage, neutrophils, etc. And so we start to be interested on this, especially because, this was shown before back in 2005, also by the lab of Van der Berg's lab, and then later on by Volker Osler. So this, we were not so much interested until we really got an observation that was really puzzling for us. So we were studying for a different reason. We were studying many host factors that allow the parasite to replicate so well. And we discovered one particular enzyme that is himoxygenase 1 and is responsible for degrading heme. And basically, when we had this, we realized that a lot of cells surrounding the infected hepatocyte were producing this himoxygenase 1 that were blocking these infiltrates. How we know that? Because when we use really, mice that were deficient for himoxygenase 1, we never saw one single infected hepatocyte intact. It was all of these kind of granulomes and fully destroyed by these infiltrates. So we thought, okay, so Plasmodium is detected while expanding the liver. Of course, there's ways to block it. So how is detected? So at that time, we had a postdoc, Peter Lille, that came to move to our lab, that did a PhD on innate immunity in Drosophila, but basically discovered something very interesting, that the parasite induces a type 1 interferon response. This was the first time that we showed that a parasite, not a virus, induced this response that was considered at that time typical of only viruses. And basically what we show is that the parasite induced this response, and these infiltrates depend on this response, because if we block the response and use mice that doesn't have the receptor to really expand this response, these infiltrates, the percentage of infiltrates surrounding these parasites, basically are not growing with time. So really, at that time, we thought, okay, Plasmodium is detected by the host. So that led to the next question that Angel Shora, a senior postdoc in the lab, started to being curious about it. So if our immune system really sees the parasite, in humans for a week, prior to reached blood stages must be that the immune system, when parasite reach blood stage, is not seeing it for the first time. It's true that it's a different stage, but it's exactly the same parasites, correct? So the question that Angel started to make it was, does liver stage preconditions the host response to then the subsequent blood stage infection? So to do that, he really used models, rodent models of infection, where we really only start infection with blood stage or start infection with the liver stage and realize what was different. And what Angel discovered is that really the pathology, the severe disease, is not only happening in the blood stage. There is kind of interplay between the liver and blood stage. And that led him to discover that indeed the liver stage really creates a kind of silent gamma delta T cell response in the liver that doesn't seem to have impact on the liver stage per se during at least the primary infection, but when you have the blood stage infection, depending of the amount of antigen that you have, basically you can go to damage and severe disease or protection from severe disease. At that time, he was very curious to understand what really was inducing these gamma delta T cells. And because we thought, okay, must be something in the liver stage, what he has decided to do it was to have parasite lines that are mutant at different, with mutants for different proteins and are deficient for different proteins. And basically, then these different proteins are important different stages of the parasite, so you can stop the parasite at different stages of liver stage development. When he did that, he found something very interesting. He found that, so basically, he had parasites that really could not traverse cells, parasites that really block here in the entrance, parasites that start to replicate but don't go further, parasites that almost fully replicate. And when he did all these experiments, only mutants incapable of traversing cells did not trigger the gamma delta T cell response. So while he's still working on that project, another project arises from an observation in different labs. So several people have shown that gamma delta T cells are really very important for sterile immunity during whole organism vaccination strategies. So what is that? Basically, okay, so then Angelo with the students are really interested in understanding if cell traversal motility is required for sterile immunity. And let me explain what is sterile immunity. Sterile immunity is really when you have a pathogen, you are infected with a pathogen, and basically you have it once and you will never have it again. So you induce an immune response that is capable of blocking any further infections. Unfortunately, that never happens in malaria in the field, with exception if we really force with pre-erythrocytic vaccines. So basically, if you use liver stage parasites, so if you inject both mice and humans with parasites from salivary glands of the mosquitoes, and these parasites cannot reach the blood because if not, they cause disease. So they can be radiated or being radiation attenuated parasites. They can be genetically attenuated parasites, as I said, parasites that are deficient on different proteins and they block a different stage of development, or either giving live parasites, fully live parasites, but really then drug treat them so it doesn't allow them to reach the blood, okay, and causes disease. If we do that, basically we know, and especially with radiation attenuated sporozoites, was shown already a long time ago in 67 by Ruth Nussenzweig, that was 100% efficacy. We had 100% sterile immunity in mice. In humans, in endemic areas, okay, basically we have 51% efficacy. So it's quite strong. So although in nature we never observe this sterile immunity, we can induce it if we use injection of high numbers of attenuated parasites. So as I was saying, what was shown is that gamma delta T cells are really important for obtention of this sterile immunity. Angelo has shown before that basically, cell traversal seems to be important for sterile immunity. So they ask really is cell traverse important for sterile immunity. So how to do these experiments? First of all, we really have some parasites, okay, that are just radiation attenuated. So we do a vaccination strategy and they are capable of traversing cells, and then they enter cells and they attempt to start replicating. They are not, because DNA has been cut to do thing, and we know that they really confer protection. And we also have parasites that are not able to traverse cells. But if we inject high numbers, some of them reach the liver in the same amounts. We know that we need 15 times more cell traversal deficient parasites to reach the liver than the ones that are totally cell traversal competent. Okay? Wild type, they are competent in cell traversal. We need 15 times more from the ones that are deficient in cell traversal to reach the liver. But even if we use 15 times more of cell traversal, while then we challenge these mice that really have been immunized with radiation attenuated sporozoites, so parasites, basically, you have protection, so the immune response have protection. These ones that really we have immunized with the cell traverse deficient parasites, we really cannot have any protection. We don't have mice that become sterile immune. The parasites still grow. There is some level of decrease when we challenge, and we are challenged with luciferase expressing parasites, so we can see the parasite establishing the liver. So this is a non-immunized mouse. So when we challenge, they have a full-blown infection. If we have these mice have been immunized with wild-type radiation-attenuated sporozoites, basically they are fully protected. But if we then have immunized mice with cell traverse deficient parasites, these mice are not protected anymore. So basically, this shows that cell traversal is required for the establishment of pre-erythrocytic sterile immunity. But Anna and Angel went further and really showed that cell traversal is sufficient alone for the establishment of sterile immunity. How they did that? So now they use a parasite, in one side the wild-type parasite as before, the other one a parasite that keeps traversing cells but never infects the final hepatocyte. So we have a parasite that is deficient in a molecule that keeps traversing cells until it gets tired, but never reach the final hepatocyte and fully replicates there. And when they did that, basically what they show is that these mice immunized with this are fully protected, as are the wild-type parasites. So basically, then we want to check what exactly was going on, how what was the lacking of cell traversal affecting what type of response. And basically what they show is that the humoral response is really not affected at all, okay? So if you really see here, what you have here is levels of IgG, and basically is very similar between mice that have been immunized with wild type or mice that have been immunized with SPECT. And we can see that initially the number of parasites that reach the liver are the same, so they are not stopped by the parasites of antibodies in circulation, and then is later on that they are not able to really, show these replication levels, and so that they disappear later on. So then go to cellular immunity. And in fact, what they show is that the cell traversal drives the establishment of these hepatics, CD8 T cell, the resident memory compartment. And they show this by flow cytometry to show the differences. And as you can see here, and I think you are seeing my pointer, I hope, that basically, the CD8 TRMs are really increased when mice are immunized with wild-type radiation-attenuated sporozoites, but not with SPECT, okay? And very interestingly, what they have done is to look for antigen-specific T cells, and they use two antigens: trap, that is a parasite that is in the surface of the sporozoite and is highly expressed in the sporozoite, and another molecule that is expressed when the parasite is developing within the infected hepatocyte. And we could see that, in the wild type radiation attenuated sporozoites, both types of these T cells and both the effector memory and the resident memories are increased in these parasites, but are very strongly decreased, especially the trap, the sporozoite specific antigen, the cells that recognize this molecule, they are fully decreased on that. So we really think this is something to do with cellular immunity and not humoral immunity. So basically, what we are saying is that cell traversal prior to infection is not only critical, but also sufficient to trigger sterile immunity under all organism vaccination strategies. Cell traversal programs cellular, but not humoral immunity. And cell traversal drives the establishment of hepatic CD8 TRM cells. Okay? But this brings something very important. How really, cell traversal shapes this level of immunity? So data that we have shows that in fact these parasites move. The parasites that really don't do cell traversal, they move as the other ones, but for some reason, as soon as they encounter a cell, they cannot go out of that cell. And they are confined on that cell, and in that cell, they expose the same level of antigen as when they are in a cell. What we think is happening is that the ones that traverse through cells, they really do something what we are calling spatial dissemination. So they traverse several cells, so they disseminate their antigens, their molecules through different cells. Okay, so basically they are extremely immunogenic, a kind of a parasite behavior that is really responsible for immunogenicity, not by increasing antigen quantity, but by distributing antigen across multiple host cells and amplifying local immune cues. So what we are really trying to say here is that we really have a parasite behavior that is an active determinant of vaccine immunogenicity. Okay, and with this, I really finish my talk by thanking everyone involved, and of course Angela and Anna that were really responsible for this story, and all our collaborators and the funding agency. Thank you very much.
Thank you, Dr. Mota. That was an amazing talk. Um, so there are a number of questions, and I'm perhaps going to take the liberty of launching into a couple myself right now. So, you've mentioned that there are sort of barriers for the parasite as it transverses across cells. And you mentioned, you alluded to Rho GTPase activity signaling being important. In the context of the hepatocytes, want to elaborate a little bit on where you feel that
Yeah,
is?
so the problem of all these aspects, as you see, there is hundreds of papers showing many different mechanisms of really things that go on. So you can imagine that, you have a parasite going on another mammalian cell, in and out, poking holes. And so obviously this causes a lot of, it's a truck entering your house and coming out on the other hand. Okay, so let's put it this way. And obviously this cause, what has been difficult is exactly to demonstrate. So many mechanisms have been shown that they activate the parasite, and we think that at least 40 to 50% of the parasite seems to be more activated when they traverse cells, being activated, being more prepared than to really invade with the formation of a parasitophorous vacuole, but it's not all of them. We have shown that the cell itself seems to be more resistant to apoptosis after going through this. As I was saying 2 weeks ago, I was with this person that is applying to our institute, and basically he was showing these results that the cell becomes really much more responsive, and it prepares the membrane to receive better the parasite. So many different these aspects. What we don't know is that are all of them important, probably, but how important they are. What is the
Yeah.
weight of each of these? And this is it's the beauty of biology, correct? Is to really take the
Tease out.
really
Yeah,
important piece of, from the noise. And I think we are starting to reach that phase. But took, a lot, 25 years, okay? So it's really a lot of.
sure. So the other point you mentioned was the interferon response, and that's very dear to my heart. Type 1 interferon response. And there's been a lot of over the years in the literature whether an interferon response is good, is bad, so on and so
Mhm.
forth. But one thing that caught my attention was your focus during your talk on the CD8 TRMs. And we know certainly in the context of a virus interferon response that CD8 T cells are really important. I'm just wondering, in the context of what you've seen, are the interferon signatures comparable? Do you see that
Yeah.
the CD8
So,
cell
yeah.
response is important?
So when we have now this particular phenotype of sterile immunity, okay, if we remove the type 1 interferon response, we, in ourselves on this specific response, we don't see a difference. Stefan Kapto in Seattle has shown that for vaccination with some parasites, in fact, the type 1 interferon response seems to be deleterious. If we take it, sterile immunity is even more
Yeah.
important. Okay, so, again, is subtle. We need to show one thing. When we show, and I think it's important also for people that are listening to us, when we show something in a rodent model, it's not that rodent models are bad. They are fantastic. I work with them, so I really think they are fantastic. But they are the tip of the iceberg. If we just really study one single model. I don't think, TRMs are going to be important for immunization of every single human. In some
Yeah.
of them, the antibody response is probably more important. In others, CD8s are going to be more important. In others, both of them are essential. And we have seen that in mice. We have mice that antibody response is really the key one. We have certain combinations and certain, vaccination strategies that the CD8s are important, others are same. So what I think is important is for us to understand what is going on, for then when we go to humans and we need then to move to human, understand exactly what we are looking for. Because if not, we are looking for a needle in the haystack, okay? So I really think we need to have a level of the diversity that is out there to see exactly how can we make something more global.
Got it. So my last question before I'll pass it on to others is, the malaria vaccines, how can we be optimistic given this range of sensitivities and so on and so forth, that they're, to be honest, ever going to be 100% effective?
Yeah, I think 100% is something that I don't think we should aspire. Okay. The world is
Okay.
not about perfection, so I'm always think that things are about imperfection. Okay. But yes, we had a vaccine that was 30% protection, so that is not good at all. Okay. And
Yeah.
this vaccine, although, has been there, I would say for more than 15 years or something, it was only during the pandemic that the WHO decided to license the vaccine and, and really put it out there. And the reason was because we were during a pandemic and it was better to have something than nothing. Okay. So when the
Yeah.
numbers start to increase again with non-governmental organizations not being in place, all these type of aspects. Of course, there is also the dangerous aspect of it that, yes, we have been educated populations to show that if you take your kid to vaccinate, they are protected. And now it's just 30%. So 30% is really on that level that is not really very protected, correct? And with all these worries about vaccines and everything that unfortunately are reappearing out of nothing, this is something that we need to be very careful how to explain to people and to make them understand. Because as scientists, we are trained to have doubts, and it's no problem for us to have doubts, correct? But for the general population to have a doubt is really problematic. Okay, so it's something people are not trained to have doubts. Doubts creates anxiety and creates is problematic. So saying this, as soon as it was introduced, because of course it was tested many different aspects, it was realized that something that in fact we knew for many decades, but sometimes we tend to forget. So many vaccines in the 60s, when they were provided in Africa, really, when they are provided during the malaria endemic areas, they are not very effective. This was described before, okay? And so now they realize the same, that probably, this vaccine becomes better. It's not suddenly becomes 100%, but becomes better when, it's just introduced during the dry season prior to the raining season. And so there is something also on the, endemic areas during malaria season that probably doesn't allow the immune system to respond in the same way. So all these is very interesting, correct, to know as well. So they are improving. All organisms vaccines, of course, many people are skeptical and doubt that at one time there will be the possibility of taking parasites, inject people with parasites, this and that. But humans are really, something very
Complex.
special, correct? So who knows?
All right, and I'm just going to ask the very last question, which was, again, you focused on the CD8 resident memories in the hepatocytes. If you look across the animals, can you see them in any other particular compartments that sort of seem to be having a role?
Yeah. So the TRM seems to be really the ones that are really, really important. And for until now, is some specific antigens and the trap that is a parasite in surface of the parasite seems to be really very, very important for what we are studying, okay? We should say that if we take gamma delta T cells, there is no sterile protection. We don't know what is still the connection. So we have now these several dots. We know that cell traversal also in this response on it, but we don't know how the dots connect with each other. Okay, we don't know if this is upstream, exactly what is going on there. But yes, there is several of these dots still that they don't have connections.
Thank you very much. Well, in the interest of time, I think I want to thank you on behalf of everybody who's here right now. We have, 150 rooms with people attending, which, many of these rooms have way more than just one individual. So it's been really exciting. And as I mentioned, over 1,700 registrants around the globe, you in Portugal, me in Toronto, everywhere. So thank you very, very much.
Thank you so
If you
much. It
have a
was my pleasure.
moment, if you can have a look at some of the questions in the chat,
Of course, I
that
will
would
do
be
that.
great. And thank you again for an inspiring talk, and we appreciate the years and efforts from your group. Thank you, Dr. Mota.
Thank you. Thank you.